And then their link actually might not let them join right until 4. But also just so you know, this is recorded and I'll explain that to the students. But just for your own information, this is being recorded for them as well.
Okay.
Awesome. And it looks like we're actually getting some students coming into the room already.
All right.
Well, welcome everyone. And we'll take a couple minutes here, just kind of let everyone get on into the room and get settled. My name is Christian Burnett. I am an Assistant Dean of Admission with the William & Mary Undergraduate Admission Office. And I am joined with the wonderful Professor Coleman today. I'm gesturing this way because that's where he is on my screen. So I apologize if it's the opposite for you all. But again, we're going to wait just another minute or two here, let all of our students come on into the room and get settled before we start today's mock lecture. Thank you so much for volunteering your time with us, Professor Coleman, and thank you so much for our attendees who are checking all things William & Mary out during our fabulous Days for Admitted Students. Again, we're going to wait just another minute or two here, make sure we have everyone, make sure we don't miss anyone. And then we'll kind of go ahead and get started. Let's see. All righty. Looks like the big flow has kind of slowed down a little bit. Well, again, welcome everyone. My name is Christian Burnett. I am Assistant Dean of Admission here at William & Mary. Again, thank you all so much for signing up for today's session. This is the mock lecture with Professor Coleman, the virus in man, the good, the bad, the ugly, and the strange. We're going to be spending a couple of minutes. Professor Coleman is going to be spending a couple of minutes giving you all a mock lecture. So it's going to feel like a William & Mary course. It's going to be very similar to what our virtual students were dealing with and kind of working with this semester. And then at the end of the lecture and towards the end of the hour, we're actually going to switch over to a Q&A. So if you have any questions as Professor Coleman is presenting, go ahead and hold on to them or jot them down. Or if you would like, you can start putting them in that Q&A box down below, but just be aware that those are going to be addressed after the lecture. With that being said, I also want to let all of our students know that today's session is being recorded. So if you have to leave early or if you have to miss something, don't worry, these sessions will become available on your welcome page, which you can access through your portal. Now, with that all being said, I know that was a lot of housekeeping tips right at the front, but with that, I will go ahead and turn it over to Professor Coleman. And if anyone has any questions or concerns, I will be in the room and in the mock lecture throughout the session. So feel free to go ahead and message me using that chat box, and I can work through any technical issues with you. But with that being said, Professor Coleman, they are all yours.
All right. Thank you, Christian, and welcome to all of you newly admitted students. I guess the first thing I'd like to say is congratulations for your acceptance to William & Mary. That's quite a coup that you pulled off there. This is not an easy university to get into, but it's one that I love. Let me tell you at the front end that this is the 51st year that I'm at William & Mary. So just to give you a sense of what that means, it means that I really do love this place. And secondly, why do I love this place? And the answer is because of folks like you. I would not stay here if my classes were populated with students that I didn't care to interact with. And I love interacting with all the young men and women who come our way here at William & Mary. It's been such a privilege. I'd also like to say hi to the parents or grandparents who might be looking over your shoulders at home through this Zoom session, and congratulate them also for nurturing you along to the point where you have succeeded this far in your lives here. I know you can't do it by yourself. There have to be some people who have nudged you along, asked you if you got your homework done, all those wonderful things. And guess what? When you come to William & Mary, they're not going to be able to do that. You're going to be on your own. And it's important to think about all of that and to think about the discipline that you need to succeed in an environment like this. Faculty are a friendly bunch. I hope you'll find that to be the case with me. I love interacting with my students and helping them. We tend to push you pretty hard here at William & Mary, but we're always there to catch you and help you. I have numerous help sessions in my classes, and the classes that I've taught over the years are organic chemistry, advanced organic chemistry. But for a long time now, I've been teaching biochemistry almost for my entire tenure here at William & Mary. And the lecture that I'm about to give you on viruses and man is traditionally given on the last day of classes in my biochemistry course. This is kind of a blow-off day. It's meant to be not a lighthearted lecture, but not one that I'm really going to quiz my students on. I just want to be informative for them when I give them this lecture. So that's sort of setting the stage for the lecture. It's done through biochemistry. The other two courses that I've been teaching lately, though, are the chemistry of emotion and behavior, which is a Call 150 class, a very writing, reading, discussion intensive class. And then a senior level kind of an exit course called neurochemistry, because I'm also an affiliated faculty member in the neuroscience program. And I'm also a pre-med advisor for my entire time here. So if you're gathering some questions together for me at the end of this presentation, I'd be happy to stick around and answer your questions as best I can about any of those topics. But for right now, let me share my screen and we'll go through this, this lecture that I've prepared for you. And as you can see here, the title is Viruses and Man, the Good, the Bad, the Ugly, and the Strange. And that's me, Randolph Coleman, Department of Chemistry, Neuroscience faculty. And of course, we are at William & Mary, aren't we? Okay, so let's think about virus characteristics. They're going to have a hereditary material, either DNA or RNA. It's going to be encapsulated either with a protein or maybe a membrane if it's a little more sophisticated kind of virus. No independent reproduction. They have none of their own reproductive machinery. They have to rely on their host cell. And I've highlighted in red here, recognize specific host cells. That's very important. The pandemic we're experiencing right now, there are some specific host cells in the respiratory system that the COVID virus recognizes. But that's the theme for all of the viruses. When they infect your body, they're going to target a specific cell. Now, are they alive? I put 2 question marks there because we really, you know, what's your definition of alive? If it's an independent reproduction, then the answer is no, as I've highlighted here. So we'll just leave that question alone there. Typical virus structure is going to have either DNA or RNA, highlighted in green here, enclosed in some kind of shell. In this case, we have a protein shell and then a lipoprotein bilayer. So this is a representation of a pretty sophisticated virus. And then there are going to be some kind of spikes on the surface made up of sugars and protein called glycoprotein. And it's these guys that tend to recognize complementary structures on the host cells. So that's where the invasion of a specific host comes from. Some typical virus coatings here. They tend to be pretty symmetric, typically, and sometimes quite beautiful when you look at them under the electron microscope, quite attractive. Now, sizes. So here's a red blood cell, which is maybe not the best representation for a eukaryotic cell, but we have that. And then we have bacteria. Here's a bacterium, an E. coli bacterium. You can see the dimensions there. And then we start seeing at this resolution, we start seeing some smaller images. And if we blow it up even more down here, we see that these guys are the viruses. So if we look at the size of the viruses compared to a red blood cell, these tiny little dots right here are the viruses. Now, what's really interesting, if we think about the last great global pandemic, which was the flu virus back in 1918, the Spanish flu, which I'm going to be talking about in a little bit. At the time that pandemic occurred, the doctors and the science community had no way to know what the invading pathogen was, because they only had light microscopes available, and light microscopes could barely resolve bacteria. So there was no way they could see the invading flu virus. They didn't know what was happening back then compared to today, where we have a thorough understanding of what's going on with COVID. So there's an interesting contrast. And if we look at these viruses here, probably my favorite is this bacteriophage T4 here. I like to describe it as like a little lunar lander. It sits down on its host cell and then injects through this central tube here. It injects its nuclear material into the host cell. So that's a pretty dramatic way to invade a host cell. What's the life cycle? Well, we have attachment, and that's where the specificity comes in. The viruses have to attach to a specific host, and that allows the host membrane to kind of open up and allow the virus to penetrate the membrane, start uncoating itself, releasing its nuclear material, which is either DNA or RNA. And then we have this fork in the road. If it's DNA, it potentially could get incorporated into the host cell DNA, where it's certainly going to take advantage of the enzymatic machinery inside the nucleus to replicate itself, and then ultimately come out to the cytoplasm, where we see protein synthesis occurring. We get regulatory proteins, structural proteins. Eventually, pardon me, it's going to reassemble and then go budding off to start a whole new population of viruses. So one host cell could bud off many, many, many replicates of the virus before the host cell becomes exhausted and probably ends up dying at that point. Okay, so some good things about viruses. We have recombinant viruses, cloning vectors, gene replacement therapy. And viruses for gene therapy typically are these three. Maybe the more common are the adenoviruses and the adeno-associated viruses, which are kind of small. We can incorporate materials into these viruses and have the virus inject that material into a specific host cell. So there are a lot of things that we can do here with this kind of gene therapy. For example, we can replace a defective portion of DNA in our bodies with a proper set of DNA. So here's a new gene in red that's going to be put into the virus, which is called a vector. It's going to recognize a specific host cell, penetrate into that host cell, get into the nucleus, open up, and have this new gene be incorporated into the host cell genome, where it can then perform its function in a proper fashion and not the mutated gene that the host cell had before. So this is something that's good, okay? That's part of the good story. We can see that happening here. We can take the virus and inject it directly, or we can harvest some cells from the human and mix the virus and the human cells in a test tube, sort out the cells that have been infected with the virus and inject them back into the host cell. So this is a little more sophisticated way to go rather than just injecting the virus directly. Some viruses are capable of fighting cancer. Here is, I call it our new ally. It's the Newcastle disease virus, the Hebrew University of Jerusalem. It was kind of discovered in two places here, but it's a virus that can be adapted to fight cancer. So it was initially a virus that infected birds, but now we're using it to infect cancers. So here is this virus that's going to have a capability of fighting a cancer. And if it infects a normal cell, nothing happens. If it infects a cancer cell, then it can release the material that we've incorporated into the virus, and it can attack the cancer cell to the point where the cell ends up lysing and killing that cancer cell, and then it can spread to attack other cancer cells. So we see that happening here. We have the oncolytic virus. It won't do anything in a healthy cell, but in a tumor cell, it can replicate itself, set off some anti-tumor immune responses from our own immune system, and attack the cancer that way. So there's some nice things that viruses can do. And in fact, in my own research, I'll talk about my research at the end of this talk, but I'm looking at some cancers also. And I think in the not too distant future, we're going to be able to fight most cancers in a fashion like this, where we can harness the immune system, harness some viruses to do what needs to be done. Okay, and that brings us to the bad: enteroviruses, the influenza virus, and herpes virus. I call these guys bad. They're doing bad things to us. For example, the enteroviruses here can attack host cells that can deal with neurologic damage or respiratory damage or cardiovascular damage. In the neurologic damage, we see viruses that can cause polio, meningitis, encephalitis. And something that's come on the scene in the last 10 years is this AFM, which is acute flaccid myelitis, which attacks the spinal cord. And a lot of young people have been hit with this virus and end up in the hospital with just a general malaise that they can't do anything. Their muscles are very weak. They're having trouble really functioning. So it's a terrible thing that's happening. I put it in the bad category rather than the ugly category. This is bad enough, okay? Whoops. And then the respiratory viruses like the flu or the common cold, and tonsillitis and pharyngitis. And then we also have enteroviruses that can attack the cardiovascular system, myocarditis and pericarditis that can attack the lining in which the heart is located, for example. So that's not good. Here's a depiction, a cartoon depiction of the cold viruses, which is a rhinovirus, and the flu virus. This is influenza type A. Some symptoms along the side here: fever, headache, general aches and pains, and so on. Do you get them with the cold or the flu? Well, one big distinguishing feature is fever. You tend to not get a fever when you have a cold, but you definitely get a fever when you have the flu. A significant fever, 100 to 102, that's a pretty serious fever. Likewise, headaches. Flu causes a significant headache, but the cold very seldom causes a headache, and so on. You can look at these things: sneezy, runny, stuffy nose and sore throat, almost always with a cold, sometimes with the flu, but not always. Chest discomfort, mild to moderate, a hacking cough with the cold, but very common with the flu and can become quite severe. So there's some distinguishing features here between cold and flu. And here's a picture of the influenza virus, an electron micrograph on the left. On the right, we see the flu virus is an RNA-based protein, so its nuclear material is RNA. It's enclosed in a protein capsid and a lipid bilayer. And on the surface, it has two important features: neuraminidase, sialidase, it's sometimes called, but neuraminidase, that's the N number we want to keep in mind. And the H number we want to keep in mind is hemagglutinin. And there are any number of variations of these two surface structures, which give us the different forms of the influenza virus, which is categorized in terms of N1 and H1 through 17 or so. So here is the flu virus, which is going to invade the respiratory tract. The neuraminidase and hemagglutinin are going to discover complementary features on the cells of your respiratory tract, invade the cell, open up the RNA, ultimately use your host cell material enzymes to replicate the viral RNA. Ultimately, that's going to come out into the cytosol be transformed into viral proteins, which are going to assemble themselves and bud off from the host cell. So one invading virus can lead to many, many replicants here inside the host cell. So we see that kind of significant increase in the virus population just from one host cell. And then there's the Spanish flu from 1918, which happens to have the designation H1N1. That's from the hemagglutinin and the neuraminidase. And that's how we characterize the flu virus forevermore. There are a significant number of different H's and a significant number of different N's. And each year when the flu comes around, we're never sure which version of the flu it's going to be. So the manufacturers of the flu vaccine kind of second guess what's going on and make some predictions. And also in the flu vaccine, you have a number of variants. So your immune system is going to be protected against a number of them. But here, this 1918 Spanish flu, globally, 500 million people were infected. 25% of the US population was infected and led to 675,000 deaths. So right now with the COVID pandemic, we're sitting at 560,000 deaths right now, and it's not over yet. People need to keep wearing masks. Okay, so we don't know where that final fatality number is going to be for the pandemic we're experiencing right now with COVID. And we're hardly seeing any flu this season because everyone is wearing masks and protecting themselves from COVID, which is the same thing you need to do to protect yourself from the flu. Okay, I'm showing virus sizes again here just to point out that when we look at the Spanish flu, the medical community didn't know what they were dealing with. Now, here are some numbers from the 2019-2020 flu season. I didn't bother getting numbers for the current flu season because we're hardly seeing any flu. But in 2019-2020, you can see that the CDC estimated about 39 million folks in the United States contracted the flu. 18 million had medical visits, 400,000 hospitalizations, and 24,000 deaths in the 2019-2020 season. Okay. The flu vaccine composition that season was a trivalent vaccine that had these compositions. You notice H1N1, H3N2, and then those are type A flus, and then a type B had this version. And there was also a quadrivalent vaccine, which had all three of the above. And then in addition, another type B flu. So depending on the flu vaccine that you received, you could have gotten a quadrivalent that had all four of these compositions that would really cause your immune system to protect you against all of those. So that's kind of nice to think about here. Then we got the herpes virus. By age 2, virtually every human has been exposed to herpes simplex. This is the virus that typically leads to cold sores of the mouth as the most common infection characteristic. And let's think about that for a minute. Cold sores. Well, it means you got a cold. And it means that when you got a cold, your immune system isn't up to par, one way to think about it. And if your immune system is suffering a little bit because you have a cold, that's kind of an opportunity for the herpes simplex virus to do its thing. So it's kind of taking over. It's sort of a sneak attack of opportunity. When you're suffering from a cold, this virus can start replicating quite easily. It typically shows up around your mouth and produces a cold sore. Now, some interesting research that just come out recently that shows a strong correlation between individuals that have suffered herpes simplex virus and Alzheimer's disease. So the other major area that I do research in is on neurodegenerative diseases. You'll see a picture of one of my research groups at the end of this talk. But I have an Alzheimer's group, and within that Alzheimer's group, I have one student, a young lady, who's looking at this correlation right now between herpes simplex and Alzheimer's disease. Then we have the chickenpox, technically varicella zoster. Chickenpox is nasty because it attacks your nerves, so it can be very, very painful. Produces blisters on your skin that are the hallmark of chickenpox, and causes nerve damage that can be pretty severe, leading to some neuralgia conditions that are not so good. So the herpes virus, if you get chickenpox when you're a child, it will produce these surface skin blisters, and then it'll go dormant and hang out in your dorsal root ganglion, where it remains dormant potentially indefinitely. But later in life, in adulthood, or sometimes for older individuals, depending on your immune system and the stresses that you're feeling, you could end up producing a case of shingles in adults. And that's not nice. So here's a young lady who's showing the characteristics of chickenpox as a child. And here's a gentleman who's showing the characteristics of shingles. And these shingles are showing up on nerves and makes them very, very painful. Okay. Now we have some nice vaccines available. We have the Shingrix vaccine, which both my wife and I have received. It's a double dose vaccine, kind of like COVID. And it pretty much is almost 100% effective against shingles. So we've kind of conquered the shingles if you get the vaccine. It's really important for you to get the vaccine. So when you get older, if you've had chickenpox as a child, you certainly want to get the shingles vaccine to protect yourself against this kind of nasty outbreak of shingles. Measles. This is measles. It's an RNA-based virus. This cartoon depiction that I found is kind of a pretty picture that shows various features. It has a capsid, a matrix protein, and a lipid bilayer on the surface, so it's fairly sophisticated. It also has hemagglutinin here and a membrane fusion protein labeled F. So a nice picture of the measles virus. We can compare chickenpox and measles. I'm not going to take a lot of time doing this. I hope you're protected from the measles. There's a vaccine available. You should have it. In fact, I think to come to William & Mary, you need to have your vaccinations all up to date. And measles is one of them. Measles is very, very contagious. If I walk into a room of individuals and I'm suffering from measles, I can almost guarantee that everyone in that room is going to get measles. It's that contagious, much, much more contagious than the COVID virus. So it's bad news, and we want to protect ourselves from measles. In 2019, there were over 1,000 cases of measles reported in the United States. And essentially every one of those cases came about because the individual did not receive the measles vaccine. I know there's a big controversy in this country about should I get vaccinated or shouldn't I? I don't really want to go there. That's more of a political statement. If you're looking at me as a scientist, I'm saying the science tells you to get vaccinated. Okay, there's no reason to not get vaccinated. And you can protect yourself from this kind of nasty outbreak. So this young man here is really suffering through a bad, horrendous case of measles. Then we have hepatitis B virus. Looks like this in a cartoon, looks like this in an electron micrograph. Causes acute and chronic liver infections. And now, another one of my research groups is Parkinson's disease group. We also see, reading the literature, that there seems to be a strong connection between hepatitis B viral infections and the onset of Parkinson's disease later on. So we have some problems there. And Parkinson's disease could be due to peripheral inflammation and a weakened blood-brain barrier. So bad things can start penetrating into your brain and start affecting the neurons that control motor movement. That would be in the substantia nigra region of your brain, where the motor neurons for your voluntary muscular system are located. And we get to the ugly here. The Zika virus, rabies, Ebola, HIV, and the coronaviruses. These are all bad news viruses here. we look at the Zika virus, kind of related to yellow fever, dengue, West Nile, and Japanese encephalitis. The mosquito is the carrier of the virus, and we try to keep mosquito populations under control. But the vectors for this virus are these two species of mosquito that will spread the virus. Now, the virus is kind of a pretty virus. If we look at its coding, a nice organization of surface structures. And then the nuclear material is RNA once again. Here's an electron micrograph with the Zika viruses stained in red, so you can see the infection in this particular individual. Now, one of the problems with Zika is birth defect, microcephaly here, where the brain doesn't develop properly, the skull doesn't develop properly. Typical head size would be in the dots here, and this a child that was born from a mom who suffered Zika ended up with microcephaly. Now, you guys might be a little too young to remember when we had the Olympics in Brazil. Brazil is a real hotbed for Zika, but there were a lot of female athletes for the US Olympic team that chose to not go to the Olympics because they were worried that they would pick up the Zika virus and then become pregnant later on and potentially produce infants who were suffering microcephaly. So they unfortunately chose to sit out the Olympic game. I say unfortunately, it's sad to see an Olympic level athlete who's trained so hard, and then because of something like the Zika virus ends up foregoing a trip to the Olympics. It's pretty sad, really. Zika can produce a rash. Here's a person suffering Zika with a rash. It kind of looks like measles. The spread of the Zika virus here, I said Brazil was a real hotbed at the time. And in the United States, if we think about where are the mosquitoes most common, they tend to be in the southern states or the states bordering water, like you see here. And of course, with some air travel, Boston, New York, Philadelphia, people can spread the virus that way, which is what we found with the COVID virus as well. And we come to rabies. Rabies is also an RNA-based virus, very deadly, almost 100% certainty that you're going to die of rabies if you don't get your inoculation shots, if you've been bitten by a rabid animal. So if we look at what's happening here is a rabid animal that's biting this poor man in the leg. The virus is going to enter the tissue through the saliva of the rabid animal. The virus replicates in the muscles and then starts moving up the peripheral nervous system, ends up into the central nervous system, spinal cord, penetrates the brain, produces a fatal case of encephalitis. The individual then is going to see the virus entering their salivary glands, just like this rabid animal, and ultimately death. So only 3 known cases of survival in the United States without treatment, 55,000 deaths worldwide. Now here in Virginia, the most common carriers of rabies are the red fox and the raccoon. We see any number of cases where both red fox and raccoons are nocturnal animals. And if you see a raccoon wandering around during the daytime, that's a good sign that there's something wrong with the animal. You want to report that raccoon to animal control and let them come and take care of it. Now, if you are unfortunate and get bitten, that's not a problem in the sense that you can easily be saved, but you do have to get inoculated. So it's important to keep in mind. Otherwise, it's almost a certain death sentence if you don't get the inoculations. Then we have Ebola. Ebola Zaire is 90% fatal. So we're talking about some ugly viruses here. It's also an RNA-based virus. Shows up dominantly in Africa, and the carriers tend to be, one main carrier is the fruit bat, but also documented, we see gorillas, chimpanzees, porcupines, and antelopes have all been known to carry Ebola. It's so fatal that it tends to kind of self-contain within a village. of course, with air travel and not necessarily containment in a village, we've even seen some cases of Ebola in the United States. There was a gentleman who had to be treated in the Philadelphia area, I think, who had just returned from a trip to Africa and brought the Ebola virus with him. Ebola in the 2014-2016 range was pretty bad, largely focused in Sierra Leone and Liberia and Guinea, and 11,000 people were killed out of 28,000 infected, which is not quite 90%, of course, if you do your math there, but it's bad enough. It's a whole lot worse than the COVID virus that we're facing right now, where the fatality rate is in the ballpark of 1% or so. There was an outbreak in 2019 in the Democratic Republic of the Congo that had somewhat greater than 1,000 cases reported with more than 700 deaths. So that's, like a 70% death rate. Pretty bad news. Then we come to HIV. That's a retrovirus carrying RNA. And global estimates show over 36 million people that were infected through 2019. Here are some projections by the World Health Organization. They're hoping to keep HIV-related deaths under these target numbers for 2020 and 2030, but it's still pretty nasty thinking about it. HIV-related deaths, the target for last year was less than 500,000. We think about COVID at 560,000. This is a terrible disease and one that can be treated. The virus is pretty neat to look at. It carries two copies of its RNA material, and it carries the enzymes that are needed to transform that RNA into DNA. An enzyme called reverse transcriptase is going to take the RNA and make DNA out of it. And then it has some other complementary enzymes that do other things as well. So the structure is pretty sophisticated. These are nice cartoon representations. Here are electron micrographs of the HIV viral particles. So what it is doing is invading a host cell. We'll talk about that in a moment. The reverse transcriptase is going to make DNA, and then the DNA gets incorporated into our own genome, and it could just lie dormant there for a very long time. And then at some point it might be triggered to replicate, and it'll replicate and produce many viruses that'll bud off the host cell. Here we can see an interesting sequence of electron micrographs that actually show an HIV virus budding off of a host cell. Kind of neat to see that that's been captured here. Now, the host cell for HIV are the cells of the immune system, specifically this central T4 helper cell. It's critical to the immune response. As you can see from this diagram, it's playing a central role in the immune response that our bodies will initiate. And if we harm that T4 helper cell, that kind of destroys your whole immune system. And that's exactly what's happening. Here are color-enhanced HIV viruses sitting on a host T4 cell, just to show that kind of attack on the host cell. Now, I said the genetic material for the HIV particles transformed into DNA, embedded into our own genome, and it can lie dormant. And typically it might lie dormant for up to a decade. So here we have a 10-year period where HIV isn't doing much of anything. So we have an acute phase when the individual first got infected, and we see some killer T cells and helper T cells and so on that have been generated, and antibodies to HIV that are produced. So the immune system was doing a nice job here, but then suddenly something happened and the whole immune system came crashing down. The HIV particles replicated rapidly, and we ended up with AIDS, which is acquired immune deficiency syndrome. Your whole immune system became deficient at that point. So what in the world happened? What's going on that this would happen? And the answer is probably some stressors. stressors can lead to cytokine formation, and that can lead to trigger in the nucleus of the host cells that will cause the virus to replicate. So latent virus activation is one area that's being studied to try to better understand what's happening. If we can prevent that latent virus from ever being activated, that would be nice. And we have some sneaky ways to do that, which I'll talk about in just a moment. Now, some individuals can resist HIV infection, and that's because their host cells have defects in the surface structures, so the HIV particles can't recognize the important host cells. So in this case, a genetic mutation in some surface structure production is protecting individuals from HIV, which is kind of interesting to think about. We sometimes think, or probably mainly think that, well, genetic mutations aren't so good, so why would you want to have one? Well, in this case, it can protect you from HIV. Now, some drug targets circled in red here, drugs that can help block the virus from entering the host cell. Drugs that interfere with reverse transcriptase, that's a great target because you and I don't use reverse transcriptase. So if we have a drug that can kind of mess up reverse transcriptase, that's going to prevent the virus from producing DNA. Some drugs can effectively work on the enzymes that lead to the production of new viruses, the building of new viruses, and the budding off of viruses. So some neat drug targets there. For a while, there was a combination of drugs that were used. Combination drug therapies are actually pretty common. In this case, it's a highly active antiretroviral therapy called HAART. But recently, individuals taking this HAART treatment were showing some resistance, drug resistance, and it's kind of been put on the back burner. More intriguing are the new technologies that we have available. CRISPR-Cas9 is a great technology that can identify a stretch of a gene that is the target, produce some RNAs that are complementary to the target, and the Cas9 is going to act in a scissors-like fashion and snip those genes, cut them apart, and they're no longer functional. So this latent DNA that the virus implanted in our genome can be destroyed. We just target that latent DNA and we destroy that portion of the genome, and the virus can't replicate. So that's pretty cool. Then we come to the coronaviruses. Coronaviruses are so named because the spike on the surface looked like a crown, thus coronaviruses. There are three now that we want to think about: MERS, SARS, and then more recently the SARS-CoV-2, which is the COVID-19 that's presently causing the pandemic. Now, the MERS virus is a Middle East respiratory syndrome virus, where the camel seems to be one of the main vectors for carrying this virus. It's showing up mostly in Saudi Arabia or the Arabian Peninsula largely. And it has spread to some extent to Britain and the European countries outlined here. Death is about 50% in the recorded cases. And it was first detected in 2012. So this is a relatively recent virus that's been picked up on. So it's called MERS, Middle East Respiratory Syndrome. And SARS is severe acute respiratory syndrome. Fatality rates for just a SARS infection, 8 to 10% in children, greater than 50% over age 60. We see that same thing being true for COVID, maybe not for children so much, but elderly individuals seem to have taken the brunt of the deaths in this country, unfortunately, and they were the first portion of the population to receive the new COVID vaccines. SARS versus MERS, they both do the same thing internally. SARS recognizes an ACE2 receptors. The MERS virus recognizes a DPP4 receptor, but once they penetrate the host cell, they kind of do the same thing. They're both RNA viruses. they end up producing new proteins from that RNA. So we have these messenger RNAs that got built. They're going to be sent to the rough endoplasmic reticulum, where the ribosomes are going to generate proteins, and ultimately those proteins are going to get repackaged into new versions of the virus. So that's what's happening with SARS and MERS. And we come to the COVID-19 originating in Wuhan, China. It's officially called SARS-CoV-2, but it's been named COVID-19, first discovered in 2019. It is an RNA-based virus, and the spike glycoproteins are really critical. It's these proteins that the vaccines are producing antibodies to bind to these spike glycoproteins and prevent the virus from infecting host cells. It's believed that the virus spread to the human population largely through the food chain. In China, there are many places where bats are considered a delicacy, or the pangolin, this mammal species here, is also a food source that can be an alternate source, and in one way or the other, it spread to humans. Now, that's the version that is believed to be true. The science seems to be saying that, although recently the former director of the CDC said he really believes that the virus was developed in the virus laboratory in Wuhan and somehow escaped that laboratory. So we might never know the ultimate truth. But if we look at the genetic makeup of the virus, it seems to be more related to what I'm showing you here. So the primary host was the bat. People eating bats, not so good, or pangolins, and we end up getting the virus in a human host, and it spreads to other humans. And then we have some therapeutic strategies. Remdesivir, which I'll talk about here in a minute, prior to vaccines, was one important therapeutic drug that helped to fight the viral infection. Now, if we compare COVID-19 symptoms to the common cold and to flu and to allergies, it's kind of hard to distinguish. Right now, the Williamsburg area is suffering under a significant pollen attack. So individuals that suffer allergies from pollen are getting the sniffles, maybe a dry cough, something like that. You see, it's pretty common in COVID. So it's hard to know unless you actually get a COVID-19 test to see if you've been infected. It's hard to distinguish among these four conditions. The COVID SARS-CoV-2 recognizes an ACE2 receptor. This is angiotensin converting enzyme 2, which is a molecule that is important for high blood pressure. The receptors are on a number of important cell types, and one cell type is in the lungs, unfortunately, so this respiratory virus can attack lung tissue. It gets uncoated. The viral RNA is going to undergo translation and produce a lot of proteins from different messenger RNAs coming off the genomic RNA. We see that translation of those messenger RNAs into new proteins, the proteins ultimately get reassembled, and the other materials get reassembled into new virons, and they go budding off. Now, COVID tends to attack the lungs. That seems to be the most significant organ that is attacked. Although we're now seeing problems with kidneys and spleen and the brain. The brain is of real interest to me because of the research that I do. In any event, we mount an immune response, and the immune response can potentially stave off the attack of the virus, but sometimes we have an overactive immune response, and we end up producing something called a cytokine storm from too much immune activity, and the cytokine storm can end up severely damaging the tissue that's been infected. And we've seen a number of cases where individuals hospitalized from COVID-19 are undergoing lung transplants because their lungs essentially have been destroyed by this overactive immune response. Okay, some treatments that could work with COVID. Antivirals, and remdesivir is an important antiviral. I'm going to talk about it a little more. Anti-inflammatories, dexamethasone is a steroid that can work. Famotidine is something that's been used to help individuals fight gastric distress. It's an anti-inflammatory. Those are both useful. And then antibody treatments, either injecting antibodies that have been harvested from an individual who suffered COVID, or now we have vaccines. So we have a vaccine, which is a much better way to go than a blood plasma transfusion, which would give you some antibodies. But the antibodies that the vaccines are producing are going to recognize those spike proteins, surround the virus particle, and prevent it from infecting the individual. So that's the way they're working. Now, early on, there were many types of vaccines that were being developed, and some of them still are being developed. The main ones that are now being produced are messenger RNA type viruses. And I'd like to throw in a plug for William and Mary here, because a young lady who did research with me back in the 1980s went on to become one of the world's experts on messenger RNA in general. And she was hired by Moderna not too many years ago, and she led the Moderna messenger RNA platform that ultimately produced the Moderna vaccine. So the Moderna vaccine had as its outcome some growth from a student from William and Mary and the William and Mary chemistry program. So how about that, folks? Kind of an interesting little tidbit there to think about. So the messenger RNA vaccines that we're taking, the messenger RNA is going to produce the spike protein, just the spike protein, not the whole virus. And that's going to lead to an antibody response, an immune response producing antibodies to that spike protein, which can then attack the real virus and prevent the virus from infecting. And this just shows that whole sequence. I really don't have time. I'm looking at the clock here. We're kind of running short a little bit here. I do want to show you one more slide here about remdesivir. This is what you would take in a pill form. When it hits the acidic part of your stomach, this linkage right here is broken. So the active ingredient is this species here after you've swallowed the pill. And what remdesivir does is it blocks the RNA replication that would be necessary for the virus to reproduce itself. So it's capable of binding to the ribosome and interfering with the ribosome's ability to produce new protein. So that's how it's working. And if we look at the scenario here, two individuals at home. Subject A has COVID-19, but no symptoms. He or she rides the train, goes to work, comes home. Some individuals at work that were in proximity to A are potentially now going to have problems. Individuals that were more social distance, maybe not so much. A finally wakes up with a fever, reports symptoms, told to stay home, gets a positive test for COVID-19, and now all these individuals, E, F, and G especially, need to be self-isolating and tested for COVID. Individuals H and I at work have a lower risk of contact, but still should be careful in the symptoms that they might pick up. And then we need to decontaminate the home, the train, and the workplace to get things back to normal. So it's a real mess, isn't it? Okay, and that brings us to the strange. I hope you don't mind me ending on a lighthearted note here when we're talking about such nasty subjects, but here's the strange. In an age with so much technology at our fingertips, it's only a matter of time until when I talk about technology at our fingertips, look at what we're doing right now, folks. We're having a Zoom session. You guys are on your technology, I'm on my technology. We all have this technology going. What's going to happen? Here's what's going to happen. We have the potential for a computer virus to spread to humans. Haha. Okay. And then I like to point out that this Weekly World News is the world's only reliable newspaper. So we don't have any fake news here. This is a real story, right? Okay. Here's a picture of one of my research groups. Right now I have 23 students broken up into different pods of studying the different neurodegenerative diseases. I have a pod that's studying different cancers. And I happen to have two young people here, which are two of my younger grandchildren sitting with me for this picture. But it gives you a sense of who I am and the size of my research group here at William & Mary. And that's all, folks. Okay, I'm going to stop my share, come back to my image. And if you all have any questions, I'd be happy to answer them. I don't know if Christian has been monitoring the chat or not, but if you want to ask me questions, I'm here, I'm yours. Until Christian says time out, and we'll quit at that point.
I would say we have just a few more minutes. I'm not seeing any questions yet, but if you have any, please feel free to put those down in that Q&A. I'm also going to put a couple links in your chat box for you. So the first one is actually to learn more about Professor Coleman's research and what he's doing on campus. Please follow that link and that'll take you to his faculty content and kind of a bio page. In addition, if there are no questions, I'll start putting some other helpful links down in that chat box. The first link that I'm going to put in there is actually to your Activate and Deposit page. So this is if you decide that you are ready to deposit, enroll, matriculate at William & Mary, that's where you'll go to do that. And that's actually, oops, that link isn't working. Give me 1 second while I get the right link in there. And then the next link that I'll be putting in there is actually it's just our admissions email account, so that if you have any questions or concerns whatsoever, you are more than welcome to reach out to us. We are always happy to help out in any way that we can.
Could I add one thing, Christian?
Yes, sir. Please do.
While we still have students here, I'm anticipating being in the classroom this fall. Okay. My two classes, the chemistry of emotion and behavior and my fall biochemistry class, are not being listed as being taught virtually. I actually have classroom assignments on campus. I'm expecting to be in the classroom with my students this fall. So I think you all can plan on a more normal experience coming up from what we've experienced for the past 14, 15 months. And I'm looking forward to it. You know, as much I don't mind this technology. It's nice to have one-on-one conferences with students. It works pretty well. It's convenient for everybody. But I love the classroom. I love the social interaction with my students. I like to go on walks around campus with my students, take some students to the coffee shop and you know, buy them a beverage and a muffin, and we chat about life and all that things. That's what William & Mary is all about. You know, that the learning is there, the rigor is there, but also these other nice opportunities need to be there. And we're missing out on all of that right now. And I'm looking forward to coming back on campus. I've been teaching out of my home office where I am right now for the last 15 months. And, you know, it's getting kind of old for me, and I'm sure for you guys too. So I'm looking forward to seeing you folks this fall.
And actually, that's a great segue to our first question. So hopefully seeing these students in particular, do you teach any first year courses or kind of introductory courses, or would students need to progress through the majors a little bit before they get to take a class with you?
No, the chemistry of emotion and behavior is a freshman level seminar, open to anybody, no prerequisites. You don't even have to be interested in science, okay? I'm going to tell you all about the brain, but you don't have to be, you know, you don't have to be a neuroscience major. You just have to be interested in the subject. And it's so popular that I teach it every semester. So it's always going to be available to first year students.
Awesome. Well, it looks like that is all of our time. I just put the link down in that chat box for everyone's Activate and Deposit page. So if you feel so inclined, please feel free to do that. And then the last thing that I'll throw in here is our admission email, so that if you have any questions or concerns whatsoever, please feel free to reach out to me or reach out to our team and we'll get back to you. But thank you all so much for spending your afternoon with us. We really appreciate it. And thank you so much to Professor Coleman for volunteering his time to help show everyone what it can be like to take a William and Mary course. Again, if you joined us a little late, don't worry, this session is being recorded and it will go up on your welcome page. So through your applicant portal, you'll actually be able to see all of the recordings throughout all of our programming and events. But with that being said, I will go ahead and end it there so that you all can get a break and before you go and do your next programs and sessions that we have coming later today. But again, thank you so much. Congratulations on your acceptance, and we hope to see you on campus this fall. Have a great day, everyone.